Hello everyone, welcome to this symposium session, Sequence and Music Processing in the Brain. My name is Yidu, I'm the chair of this session. Music is a human universal involving perceptually discrete elements organized into hierarchically structured sequence. There is ongoing scientific endeavor in studying how the human brain processes music using state-of-art neurophysiological and neuroimaging techniques. This symposium focuses on the brain mechanisms underlying the perceptual and emotional aspects of musical processing, including the functional organization of sequence structure, cortical tracking of musical rhythm, predictive coding of musical phrasal structure, and emotional responses during music listening in normal and special populations. They have 5 wonderful speakers. The first speaker is Professor Nadine from Zhejiang University. The title of his talk is Sequence Structure Processing in the Sensory and Ocular Motor Systems. Let's welcome.
Hello, everyone. Welcome to my talk. My name is Nadine, and my talk is entitled Sequence Structure Processing in the Sensory and Ocular Motor System. We live in a dynamic world, and we saw and hear all kinds of dynamic stimuli, such as speech, music, and dynamic visual scenes. For example, when we hear someone speak, we hear a sequence of words, such as Tom went on vacation. When we hear the sequence of words, we have to put them together to understand the meaning of a whole sentence. Furthermore, when we put together the words, we have to follow certain rules. For example, here, we have to put together on vacation first into a prepositional phrase, and then put together this phrase with went, and then put together Tom with the rest of the sentence. Furthermore, when we put together two units together, usually one is more important. For example, in on vacation, vacation is the more important word, also called the head of the phrase, which we should pay more attention to. And similarly, when we put together the subject and the predicate of a sentence, the predicate is the head, which we should attend more to. So when we process a sequence, when we integrate information, we have to consider the structure of the sequence. And often we have to attend to different units to a different amount. To probe how the brain integrates sequential information over time, a few years ago, we did the following experiment. In the experiment, we used computers to synthesize a bunch of syllables, and all the syllables were independently synthesized so that there's no relationship in terms of the acoustic properties between these syllables. And we concatenated the syllables into a sequence so that every 2 syllables construct a phrase, and every 2 phrases construct a sentence. And to facilitate the analysis of the experiment, we normalized the duration of all syllables to be the same, to 250 milliseconds. And there's no acoustic gap between any phrase or sentence, so that all the syllables were presented at a constant rate of 4 Hz. Correspondingly, the phrases were presented at a constant rate of 2 Hz, and the sentences were presented at a constant rate of 1 Hz. And we had both Chinese sentences and English sentences. And here I'm presenting the neural response to Chinese sentences recorded from a number of native Chinese listeners. And the neural activity is recorded using MEG. And clearly in the MEG response spectrum, we saw 3 major components, which are at the syllable rate, the phrasal rate, and the sentential rate, showing that the brain indeed concurrently track all the 3 linguistic levels. Furthermore, when we present the same Chinese sentences to English listeners who did not understand any Chinese, we found that their brain could only track the lowest level, the syllables, and they could not mentally construct higher level units such as phrases and sentences, and there is no response related to these two levels. By comparing the responses from the two groups of participants, we can see that only when we have the tacit linguistic knowledge, we can group syllables into larger units such as phrases and sentences. And all the different linguistic levels were represented by neural activity on different timescales. And we also recorded the neural responses to such sentences using intracranial EEG, and we found that the phrasal rate response and the sentential rate response were generated from widespread cortical networks containing both the temporal lobe and the frontal lobe. To summarize this part, we found that when we listen to speech, our brain constructed multiple levels of neural representations corresponding to multiple levels of linguistic units, such as syllables, and also words, phrases, and sentences. And the different levels of linguistic units were represented by neural activity of different timescales in different cortical networks. And a few years later, we tried to replicate the experiment using EEG. And indeed, we found that the results could be replicated using EEG. However, we had a new finding that there is also a response tracking the sentences in the EOG signal that we recorded. We recorded the EOG signal since there is a large EOG artifact in EEG, so that we have to record the EOG signal to cancel such artifact. However, we found that in the kind of artifact that we record, there's this component tracking the spoken sentences, which is really weird because in the experiment, the listeners were only listening to the sentence. They didn't see any meaningful thing and they actually had their eyes closed. However, somehow their eyes seem to be tracking the spoken sentences. To further analyze what kind of eye activity is tracking sentences, we did an additional eye tracking experiment. We found that for all the eye tracking measurements, such as saccades and blinks and pupil dilation, only the blink is correlated with the sentential structure. In other words, when we analyze the blink sequence, we see that the response also track the sentential rate, similar to the vertical EOG signal, which means that when we listen to sentences, we tend to blink in sync with the sentences, which is still weird because we use our ears to listen, but why do we move our eyes when we listen to sentences? And a hypothesis that we came up was that maybe the ocular activity is modulating temporal attention, because a large number of studies have suggested that the motor system is involved in modulating attention. And of course, attention is also important when we process sentences. And the ocular motor system also plays a fundamental role in visual attention. And we hypothesize that maybe it can also guide temporal attention during speech processing. To test this hypothesis, we did a new experiment in which we presented the same set of sentences, but gave the listeners a different task. In the first task, the listeners only have to attend to the first syllable of a sentence. And in the second task, the listeners were asked to attend to the third syllable in a sentence. And to make sure the listeners follow our instructions and attend to only one syllable in a sentence, we gave them a very challenging task. In the task, at the beginning of a trial, they see a phoneme visually, and then they have to detect whether any of the syllables they attended to start with this phoneme. This is a challenging task, but the participants can do the task after some training. And because the task is challenging, they have to focus on the syllables that we ask them to focus on in order to have a high performance. And we hypothesize that if any kind of activity tracks the attentional focus, this kind of activity should be different in the two attention conditions. And more specifically, in this experiment, the activity should show a 180-degree phase difference between the two conditions. And this is actually indeed what we found in the EOG signal. We found that when the listeners attend to the first syllable and the third syllable, their EOG activity oscillate out of phase. So this result confirmed our hypothesis that indeed EOG activity or blinks reflect the temporal distribution of attention. And following this question, we ask another question, that is whether this mechanism is specific to speech processing. And to test this hypothesis, we presented to listeners simply a sequence of tones. And we asked the participants to imagine that all 4 tones construct a group, and their task is to either attend to the first tone in a group or the third tone in a group. And they have to detect whether the tone they attended to is replaced by a frequency modulated tone. In every trial, there is a frequency modulated tone, but the listener should only judge whether their attended position showed a frequency modulated tone. And we found a very similar results, that when the listener listened to the tone sequence, there's also a EOG response and blink response tracking the perceived 4-tone group at 1 Hz. And when the listeners attended to different tones, their ocular activity and blinks oscillate out of phase. And similarly, we also tested whether the same phenomenon holds when we process a visual sequence. I will not go into the details here, but we saw the same phenomenon when viewers attended to different position of a sequence. Their blink pattern depends on their attentional focus. Finally, we ask, when do they blink when they process a sequence? In the visual condition, it's very clear that they tend to blink about 700 milliseconds after the time moment when they attend to. And surprisingly, it's also what we found in the auditory sequence condition. So whether we listen or watch a sequence, we tend to blink about 700 milliseconds after a time moment when we attend to. So to summarize the whole study, we found that the motor system truly engaged in sequence processing, and its role is to modulate the distribution of attention in time. And specifically, we tend to blink about 700 milliseconds after the time interval when we pay more attention. Okay, thank you everyone for listening, and I also want to thank my lab members and all my collaborators. Thank you, bye-bye.
Second speaker is Professor Yunnan and her student Xiangyun Meng from Beijing Normal University. The title is Theta Oscillation Predicts Neural Entrainment to Musical Reason. Let's welcome.
Hello everyone, my name is Meng Xiangyun. It's a great honor to share with you the research of our group. The title of my presentation is The Development of Internal Theta Oscillation Predicts Neural Response to Musical Rhythm. In language processing, the brain can not only track the syllable frequency, but also entrain the frequency of higher-level structures, such as phrases and sentences, reflected by spectral peaks at corresponding frequencies. The neural activity is dependent on language comprehension. Similarly, in musical rhythm processing, the paradigm is to ask participants to listen to rhythmic beats while recording their EEG. In this study, they found that the 2.4 Hz beat elicits a sustained periodic EEG response tuned to the beat frequency. Then they asked participants to imagine a binary or a ternary meter structure on this beat. They found that meter imagery elicits an additional frequency tuned to the corresponding imagery metric of this beat. After removing the external stimulus, they found that the entrainment response remains. We can see the brain can entrain external stimuli. However, the entrainment response is not completely invoked by the external stimuli. Dueling proposed the oscillator model hypothesis. The oscillator refers to a system that generates sustained rhythmic behavior by itself. Instead of passively tracking the stimulus envelope, there is internal oscillatory synchronization. We're just wondering, without external rhythmic stimuli, whether there is an internal oscillator which plays a critical role in the brain mechanisms of musical rhythm processing during development. So we use spontaneous resting state neural oscillations. Classically, delta and theta power decrease with age. Alpha and beta power increase with age. On the other hand, gamma power increases at first and then decreases as children grow older. Resting state neural oscillations possess a great value. In our previous study, they could serve as sensitive biomarkers that predict vocabulary development in school-age children. In this study, we recorded resting state EEG and rhythm task EEG to explore the relationship between them. In Experiment 1, we included 46 typically developing Mandarin Chinese children from a longitudinal program. They were tested at ages 7 and 9. We also coded their music training experience. We recorded eye-opened resting state EEG data for 5 minutes at both ages 7 and 9. For rhythm task, we recorded a passive audible paradigm at age 9. The stimulus was a 3-note bar with the first note at a strong beat. The standard duration of each note was 500 milliseconds. For the small event, the duration of strong beat was 600 milliseconds, and for the large event, the duration was 660 milliseconds. We also collected active behavioral rhythm discrimination tasks and nonverbal IQ, working memory, and musical experience as control variables. Results showed that no significant developmental change for all frequency bands from age 7 to age 9. Rhythm behavior performance showed a significant condition effect that children performed better on large events than on small events. MM response showed significant main effects of condition and area. MM was larger for small event than for large event, and larger in frontal and frontal-central areas. Correlation results showed that resting theta oscillation ranging from 4 to 8 Hz at both ages 7 and 9 were related to musical rhythm MM for large events. Hierarchical regression analysis showed, after controlling for general cognition and musical experience, resting theta oscillation at ages 7 and 9 still contributed to musical rhythm MM at age 9. In experiment 1, we didn't find the developmental change of resting state neural oscillations, which may be due to the small age range. The distribution of the MM component in frontal and frontal-central areas is consistent with previous studies. Resting state theta is related to musical rhythm MM. Most importantly, theta remained a key predictor of rhythm processing after controlling for nonverbal IQ, working memory, and musical experience. Now we know the internal oscillator plays a role in the brain mechanisms of musical rhythm processing. Is there a causal relationship between spontaneous neural oscillations and musical rhythm? Does brain oscillations affect rhythm, or in reverse, does music ability promote brain development? Here we had 52 children and followed up these children till age 11. All of these children completed resting state and rhythm perception EEG recordings. Based on experiment 1, we focused on theta oscillation and MM for large events. Behavioral measures follow the procedures mentioned in experiment 1. Results showed a significant decrease of theta power from age 9 to 11. Rhythm behavior performance showed significant improvements in detecting both small and large events from age 9 to 11. For MM response to large events, no significant age effect was observed. Correlation results showed a significant correlation between resting theta oscillation at age 9 and MM for large musical rhythm events at age 9. Theta oscillation at age 9 significantly predicts MM at age 11. And in reverse, MM at age 9 significantly predicted theta oscillation at age 11. Cross-legged panel analysis showed a significant path from resting theta at age 9 to musical rhythm MM at age 11, whereas the path from musical rhythm MM at age 9 to resting theta at age 11 was not significant. Hierarchical regression analysis showed a similar pattern. After controlling for general cognition and musical experience, both theta oscillation at age 9 and theta power decrease from 9 to 11 could predict musical rhythm MM significantly. After controlling for autoregressive MM at age 9, theta oscillation at age 9 and theta power decrease could also predict musical rhythm MM. However, when predicting theta, only MM at age 9 was significant. Next, let's move on to discussion. In experiment 2, theta power decreased from age 9 to age 11. In our recent study, the resting brain oscillations from all three assessments revealed the developmental change of neural oscillations, which is consistent with previous studies. No developmental effect of MMN from age 9 to 11 was found, but we found significant improvement in behavioral performance, which means reason perception ability still developed from age 9 to age 11. Finally, both cross-legged panel analysis and regression analysis showed a significant path from resting theta to rhythm MMN, whereas the path from rhythm MMN to resting theta was not significant. So theta oscillations may be the physiological basis of rhythmic development, which suggested a causal relationship. There is the spontaneous internal oscillator that exists in resting state, independent of stimuli or tasks, which participate in rhythm perception or contribute to the brain mechanisms of rhythm processing. We provide evidence for the oscillator model hypothesis. To sum up, resting theta oscillation plays a significant role in the development of musical rhythm perception, possibly serving as an important precursor to the development of neurorhythmic processing. And we support the oscillator hypothesis. That's all. Thanks for your listening.
The next speaker is myself. The title of my talk is Music Listening as Time Travel, Prediction on High-Level Musical Structure Underpinned by Phrase Processing of Auditory Oscillation. Hello everyone, it's my great pleasure to present our recent work on prediction of high-level musical structure underpinned by phrase processing of auditory oscillation. This work is done by my student Yiyang Wu, Si Yu Lai, and my wonderful collaborator, Dr. Xiangbin Pan. Same as language, music is organized into hierarchically structured sequence with perceptually discrete elements. Previous research in speech neuroscience has identified cortical tracking of hierarchical linguistic structures at the syllable, phrase, and sentence levels in speech. So can we find similar neural entrainment to hierarchical structure in music? Previous research in music neuroscience, however, revealed cortical tracking of musical notes, which is modulated by musical expertise. At a higher level, we also found cortical tracking of musical beat and meter. But how do they process music at more abstract structural levels, such as at the level of phrase? In a study by Stephen Koch, they manipulate the final chords in a musical phrase, and they found an early negative and a late positive ERP components in response to regular chords at a phrasal boundary, which indicate processing of a hierarchical structure with nested non-local dependency in music. However, this only provides offline and indirect neural index. It remains largely unknown how does the brain learn and online establish high-level musical structure during music listening, which is a core of attention and pleasure in music. So the current study first aimed to reveal neural evidence for online segmentation and prediction of hierarchical musical structures at the note, beat, and phrasal levels. This is to replicate findings in Dr. Tan's latest work, which is already published online on bioRxiv. Lots of details about data analysis and experimental design can be found in this paper. And our second purpose is to look at how does repetitive listening to music, which is quite common in our daily life, modulate the segmentation and the prediction of musical structure. So we chose 10 chorus by Bach. They all have very regular musical structures, and we did two types of reversal manipulations. One is local reversal. So they locally reverse the beats in the middle of musical phrases while keeping the onset and the offset beat intact. The other one is global reversal. So they globally reverse the temporal order of beats in a musical piece. Theoretically, the harmonic progressions, which is indicated by dashed tree structure, is preserved in the original and global reversal conditions, but not in the local reversal conditions. Since the harmonic progressions enable listeners to anticipate the musical structure boundary, the neural signal should lock to the musical phrasal structure to a higher extent in the original and global reversal conditions, and to a less degree in the local reversal conditions. And in the original pieces, we can also find a phrase processing phenomenon, which means the neural signal advances faster than the physical musical structure. So here is an experimental design. We have 30 long musicians as our participants. We use Gold MSI questionnaire to measure their musical training experience. All of them have less than 3 years of formal training, and each musical piece was presented 3 times consecutively. After 3 times of listening, participants have to rate how much they like this piece. From the liking rating, although the original piece got higher rating than the other 2, there was no significant difference. And this is a 3x3 design. We have 3 musical conditions and 3 times. And each recording was done by NeuroScan 64 channels at a sampling rate of 500 Hz. And we used an off-live bandpass filter at 1 to 35 Hz. And this is the EEG analysis pipeline. We first extract the shared neural component across the participants using MCCA, which is a multi-rate canonical correlation analysis, and we select the components that explain the largest variance. And then we did a first order FFT to measure the beat and note tracking around 1 Hz and derive temporal response function and the cerebral acoustic coherence. And next, we did a second order FFT, which reveals how the power of the neural signal was modulated by the phrasal structure around 0.1 Hz. And next, we quantified phrasal phrase processing in of neural phase and phrasal boundary plane. So phase processing occurs when the neural signal advances faster than the phrasal boundary. Otherwise, phrase recessing occurs when the neural phase advances slower than the phrasal boundaries. Here is the results. Old news is consistent with previous studies. We found reliable beat and note tracking. This is results from cerebral acoustic coherence. We can find reliable neural tracking at the beat frequency and low frequency. And we look at how the beat tracking and note tracking was modulated by repetition. So we found the beat tracking did not change with repetition, but the note tracking decreased with repetition, with the third time significantly lower than the second time. And the note tracking in the original condition was correlated with musical training experience. We also directly calculate the spectral magnitude at beat rate and note frequency. And this time, we found the beat tracking decreased with repetition, with the third time significantly lower than the first time. And the note tracking showed a significant condition by time interaction, with the third time significantly lower than the other 2 times only in the global reversal condition. Moreover, we found note tracking in the original condition at the first time was correlated with musical training. Here is some news. We also adopted a method called temporal response function and neural decoding to investigate the reconstruction accuracy of musical envelope. And the reconstruction accuracy showed a significant condition by time interaction for global condition. The third time was significantly lower than the first time. For original condition, third time was significantly lower than the second time. So from the beat tracking, load tracking, and envelope tracking, they all consistently show that repetitive listening weakened neural entrainment to musical acoustic features and low-level structures. Here is some new news. Interestingly, we found EEG power at beat frequency, which is around 1.4 Hz, was temporarily modulated at the phrasal boundary. If we did a double FFT to reveal the modulation spectrum, we can find the modulation frequency peaked at 0.177 Hz, which is the phrasal frequency. So that means EEG power at beat frequency was temporarily modulated at phrasal frequency. This is a way we show the phrasal tracking. Next, we quantified the modulation spectrum at the phrasal frequency, and we found reliable phase tracking at all the conditions. And we also look at how this modulation strength was modulated by repetitive listening and condition. So this showed a significant main effect of condition and condition by time interaction. For global reversal, the third time, the phase tracking at the third time was significantly stronger than at the second time. And at the second time, the phase tracking in the global reversal condition was significantly lower than other two conditions. This means globally reversed music showed a slow build-up of phase tracking. They also investigate the correlation between phase tracking and the music trailing experience. And this correlation was significant only in the original condition, but not in the other two conditions. Lastly, we look at the phrasal phrase processing. So this showed the phrase series of grouped averaged EEG power of an example musical piece. The phase of 0 indicate where the peaks of cosine waves should be. The neural phase at the phrasal boundary in the original and the global reversal condition was advancing upwards as the phrasal structure unfolds. They fit a line between the phrasal boundary and unwarped boundary phase series. If no phase processing occurs, the slope should be 2 pi, that's the dashed line. If phase processing occurs, the slope is larger than 2 pi. The difference between the slope of condition and the 2 pi is indicated as phrasal phrase processing index PPPI, which indicate the degree and direction of the phase processing. So interestingly, we found significant phrasal phase processing occurred when listening to the original piece only once, indicating the speedy build-up of prediction of musical phase structure even in long musicians. They also quantified this PPPI index and look at how this was modulated by condition and repetitive listening. And PPPI showed a significant main effect of condition, with local reversal significantly lower than original and global reversal. What this means? This means the phrase segmentating neural component actually lag and follow the phrasal boundary in the local reversal condition, but lead and predict the incoming phrasal boundary in the original condition. If we think music listening as a time traveling, as the phrasal phrase processing occurs, the time is warped mentally. Here is discussion and summary. We found some old news consistent with previous studies, beat tracking, load tracking, and envelope tracking in music perception, and no tracking in original music is modulated by musical expertise. We also found some new news. Note tracking, beat tracking, and envelope tracking decrease with repetitive listening, indicating learning-based enhanced prediction, but weakened neural entrainment to musical acoustic features and low-level structures. Continue with new news. EEG signals at ultra-low frequency around 0.1 Hz track musical phrasal structure, providing a novel and direct neural index to examine music on line segmentation and high-level structures. Phase tracking in original music is modulated by musical expertise. Phrase tracking in global reversal music is hard in non-musicians, but can be gradually established by repetitive listening. Phrasal phrase processing is quickly established in original music, but is not modulated by repetition, demonstrating the long-term memory-based predictive coding of music, high-level structures. Phrasal phrase processing is found in local reversal music. That means disruption of harmonic progressions preclude the prediction of musical phrasal structure. Some take-home message. So our human brain can segment music streams online and establish high-level musical structures using ultra-low neural oscillation in a manner of predictive coding during repetitive listening. So many thanks to my student, collaborator, lab members, and fans. Thank you all for your attention. Our next speaker is Professor Jing Lu from University of Electronic Science and Technology of China. His talk is about the brain mechanism of alleviating anxiety by music and EEG study.
Hello everyone, today I would like to talk about understanding the effects of music on pain and state anxiety. There are lots of connections between brain and music. Lots of studies have shown music have many positive effects on the brain. Music can influence perception of effort and makes us forget we are tired. Listening to music releases dopamine in the brain. Music can improve sleep and relaxation, and so on. Our group recently tried some clinical application on music intervention. As we know, there are lots of advantages from music intervention for clinical use, such as economical, non-invasive, convenient. A survey by American Music Therapy Association showed over 20% mental health can use music intervention, and also many other diseases such as Alzheimer's disease, neurological disorders can use music intervention. But the problem is the brain mechanisms by the music intervention still unknown. Here we took pain and state anxiety as an example. First, let's talk about music and pain. Pain is a very complex experience caused by tissue damage or other noxious stimuli, and it can bring unpleasant feelings and emotions. There are some factors can affect pain perception, such as personality traits, emotions, and attention. The most commonly used intervention for pain is drug therapy, but it's a little bit expensive and with side effects. Music therapy is easy to develop with low cost. So some studies has shown the music therapy has a positive effect on pain relieving. Over 50% of patients say they would listen to music most of the time in their life. Also, over 50% of the patients thought that music was much more important for them than ordinary people. The role of music for them was mostly to make them feel better, relax, and reduce pain. Some studies show that music therapy is effective to reducing the pain, such as the study on the pregnant women, and even some studies on the critical ill diseases such as cancer. But there is little research on how to choose music, which music is more effective, and what's the mechanism behind the intervention. Here we design an experiment to study the music and the pain. We recruit 40 healthy people to ask them to put their hands in the cold water to induce the pain. And the tolerance time they can put their hands in the water was marked as the pain scale. That means the longer their hands can put in the cold water, that means they can tolerate more pain. And we asked them to listen to 3 kinds of music: the happy music, the neutral music, and the sad music, while their hands when their hands put into the cold water. And we, after experiment, we divided them into 2 groups. One is a remission group and another is a non-remission group. Remission group means the tolerance time while listening to music is longer than the pain tolerance time with no sound. That means the music can relieve the pain. The music is effective for this subject. And during the data analysis, we do the FFT for their EEG data, because during the whole process, we put the EEG device, the EEG cap on their head, and we record the EEG signal. And the FFT results showed in the remission group, when the subject listening to the sad music, their higher beta band and the gamma band of EEG showed a significant stronger than with no sound condition. So it seems the sad music take effect to relieve their pain. And we also do the correlation analysis between the spectral amplitudes and the pain tolerance time, the correlation analysis between personality tree score and the spectral amplitudes in the gamma band. And also we do the connectivity analysis, and we can show when the remission group listen to the sad music, the coherence network showed a parallel increase in connections, mainly in the prefrontal, parietal, and occipital regions. So this experience, we can find the remission group while listening to sad music showed that the O2 and P4 electrodes has a higher FFT amplitude in the gamma and the higher beta bands. This result may reflect the effects of music on cognitive functions such as pain perception and selective attention. The correlation of the personality shows the personality may play an important role in music-induced emotions. And introverted subjects, aesthetic appreciation of sad music will lead to a higher pain tolerance. And the sad music not only increases activity in the parietal and the occipital regions, but also leads to increased functional connectivity, which is associated with competition between pain perception and emotional processing. And the literatures show the changes in cognitive emotional effects could be reflected by gamma neural oscillation in parietal and occipital regions. And when listening to sad music, the enhanced connection may be related to the competition between pain perception and emotional processing. And the effect of sad music regulation pain may be related to aesthetics of sad music caused by personality. So those literatures can support our findings. Then we talk about the music and the state anxiety. State anxiety is a situation-specific form of anxiety which can be experienced by anyone. And long-term state anxiety often elicits somatic symptoms such as nervous system problem, the breathing disorder, even the risk of suicide, and so on. The commonly used interventions for state anxiety is the drug interventions, of course, and there are also some non-drug interventions, such as music therapy. We will talk here. And there are also some studies for the music intervention on state anxiety. And these studies find listening to music may also help patients with severe disease to ameliorate state anxiety and reduce the antimicrobial use. And the music intervention may be beneficial in reducing state anxiety and can be applied in pregnant women. And some studies also reveal that the neutral and happy music can work for the state anxiety. Although these studies have indicated that music interventions can relieve the state anxiety, the type of music that works best and the brain mechanism behind them is still far from clear. So here we designed an experiment, just like we did the pain experiment, to study how music can works on the state anxiety. We recruit 62 healthy students in the university, and first, during the first stage, we induce anxiety by asking them to listening to some sounds and and also watching some pictures and induce their anxiety. And after the inducement, we test, we're doing a questionnaire and test whether they have been induced the anxiety. And after that, we divided them into 3 groups, asked them to listening to neutral music, happy music, and blank stimulus individually. As a result, we can find the picture A shows all groups has been induced as anxiety, and picture B shows after the anxiety intervention, we can see the neutral music group show the greatest effect. And also we have some positive for the happy music group. And we do the power spectrum because during the all time we ask them to wear the EEG cap and record their EEG signal during the whole process. And we can see the difference between the power spectrum among the 3 groups. The power spectrum density of neutral music group showed more decrease than happy music group and the blank stimulus group. And the positive correlation between the scores of the state anxiety and the power spectral density and the occipital lobe in the gamma band in the condition of listening to music. This is the result this slide shows. And from the seed of the occipital lobe, in SO2, we did the functional connectivity. We can find during the neutral music group, the occipital lobe connect with the frontal lobe, and in the happy music group, the connectivity shown between the occipital lobe and the temporal lobe. And we also do the analysis of neutral music and personality. We do the correlation. And we can see from the results above, the neutral music had a better effect on relieving the state anxiety. The effect of happy music was suboptimal. It can also work, but the effect is not good as a neutral music. We also find the trait personality and trait coping style played important roles in anxiety intervention. Studies provided evidence for that disturbing pictures rated as high anxiety provoked activation in the occipital cortex and both amygdala. Therefore, stress-induced anxiety may be closely linked to an activity in the occipital gyrus. Through power spectral analysis, the occipital cortex emerged as a region of interest. We find that the brain connectivity between the occipital lobe and the frontal lobe increased after neutral music intervention. It suggests that the occipital lobe is a hub node in the process of music intervention in state anxiety. For the neutral music intervention, the change in brain function mainly originated from emotion, attention, and impulsive working together. For happy music intervention, the change in brain function mainly originated from the happiness emotion and anxiety trading off with each other. The anterior portion of the bilateral temporal lobe is vital in the discrimination of melodies and chords. Inferior frontal areas may also be implicated in empathy, compulsive behaviors, and changes in personality and social deficits. Higher scores on the extroversion personality trait were accompanied by less anxiety induced by neutral stimuli with neutral music and reading. In addition, coping style, which is known impact psychological well-being, may serve as a control for variables in the future studies. So let's summarize the work. According to the two studies above, we can find the sad music has the positive improving effects on pain. The neutral music has a better effect on alleviating state anxiety. The brain mechanism of pain relief by music is related to the gamma neural oscillation in parietal and occipital regions. Aesthetics experience influenced by personality traits play an important role in this process. Decreased gamma power spectral density of occipital lobe is associated with neutral music alleviating state anxiety. Emotional responses is the intermediary factor for music intervention. And these findings above are helpful for understanding music processing in the brain and have potential clinical use as well. Here are some take-home messages for the talk. So we find the emotions plays an important role in the processing of neutral music, alleviating state anxiety. So probably the music emotion state anxiety intervention loop needs to be explored in the future study. We also need to study the influence of music elements such as the rhythm, pitch, timbre, and so on. And the last, during these two studies, we recruit the healthy subjects and to induce pain and induce anxiety. In the future, we need to recruit the patients to study, to test whether this music also have the effect. So finally, I would like to thank the group, the neuromusic group in UESTC, the Professor Yao and me, and these are students in the group. And we also thank the grants support these studies. This is my talk. Thanks for your attention.
Our final speaker is Professor Chunmei Jia and Su Zhou. The title is The Autistic Brain, Loss of Global but Not of Local Processing in Emotional Music and Faces. Let's welcome.
Hello everyone, I'm Lin Shuzhou from Shanghai Normal University. Now I'd like to share a study on autism. In this study, we investigated how individuals of autism process facial and musical emotions. During information processing, we process both local and global information. Normally, we have global bias, which means when we see a forest, we see a forest, but no individual trees. However, individuals with ASD have atypical local bias. There are two theoretical accounts. One proposes that the local bias results from the global deficit. The other argues that the local bias can occur without a global deficit. The main difference between these two theories is whether ASD individuals have deficits in global processing. The global processing involves regularity extraction and integration. Previous studies on ASD global processing mainly focus on integrative processing. Few studies focus on regularity processing. Regularities can occur on different timescales, such as local and global levels. For example, in block 1, the tone sequences d-d-d-d-d interval d-d-d-d-d interval form a global regularity. In block 2, the sequences d-d-d-d-d interval d-d-d-d-d interval form another global regularity. Within the sequence of d-d-d-d-d, the fifth tone represents a local regularity violation. However, across sequences in block 2, the sequences of d-d-d-d-d represents a global regularity violation. Only one study investigated the regularity processing at local and global levels in ASD. In this study, ASD and TD controls show comparable neural responses to violations of global regularity. However, ASD saw reduced responses to local violations. This finding is somewhat inconsistent with the local bias hypothesis in ASD, and it may be due to the fact that they simply use the repeated sounds in tone sequences. Given that ASD is featured with social communication difficulties, it will be worth investigating the process of abstract emotion regularity at both local and global levels. Poor ability to understand emotion may be one of main causes of social communication difficulties in ASD. However, little is known about neural responses to emotion regularity in ASD. Because understanding facial expression is an important component of social communication, facial expressions provides an ideal case for investigating emotion regularity processing in ASD. Several behavioral and neural studies have shown impaired facial expression processing in ASD. None of them focus on neural responses to emotion regularity. Moreover, musical emotional processing is also an important part of social communication. Indeed, music has been regarded as effective in treating facial emotional disorders in ASD. So, it will be interesting to make a comparison between facial and musical emotional processing. In this investigation, we use ERP to investigate whether individuals with ASD will show abnormal neural responses to local and global emotion regularities extracted from sequential facial expressions. And if so, whether this abnormality will generalize to the musical domain. We mainly focus on 3 ERP components. It has been suggested that the mismatch negativity responds to violations of local regularity at a pre-attentive level, while the P3B responds to violations of global regularity at a conscious level. Furthermore, that negativity reflects the top-down allocation of attention for the inspection of memory traces. Given that ASD is associated with a local bias, violations of local emotional regularity will elicit the mismatch negativity in ASD. If individuals with ASD have a global deficit, violations of global emotional regularity might elicit P3B or the let negativity in TD only, but not in ASD. The first experiment used images of facial expression to investigate the neural responses to local and global facial emotion regularity. 20 individuals with ASD and 21 individuals with typical development participated in this study. The 2 groups were matched in age, education, musical training, and IQ. An emotional audible paradigm was used in our experiment. There are 5 images in each sequence. They appeared one by one with fixed intervals. There are 125 trials in each block. The first 25 trials established the global emotional regularity. The following 100 trials consisted of 80 frequent sequences and 20 rare sequences in random order. For the blocks with local deviations, the standard had 5 identical facial expressions, while the deviant had 4 identical facial expressions followed by a different facial expression. The different facial expression violated the local emotion regularity. For the blocks with global deviance, however, the standard has 4 identical facial expressions followed by a different facial expression, while the deviant had 5 identical facial expressions. As such, the same facial expression violated the global emotion regularity. For each sequence, the 5 faces were physically different, so each stimulus was constructed based on abstract regularity of emotion. Participants were informed that each sequence consisted of 5 images and that a few of the sequences might be different from the majority of these sequences. They were required to count the number of those deviant sequences and report the number after each block. The ERP were time-locked to the 5th image. We compare the ERP amplitudes elicited by standard with deviant stimuli in each type of blocks. As shown in the figure, the red indicates the deviant and the blue indicates the standard. In the early stage, local deviant images elicited the mismatch negativity in SD but not in TD. In the later stage, however, only TD exhibited a P3B and an LPC in response to local emotion regularity. The P3B and LPC were distributed over central parietal regions. Global facial emotion regularity elicited a P3B, followed by an LPC in TD but not in SD. These effects were distributed over central parietal regions. The observed mismatch negativity indicates that the autistic brain can detect violations of local emotion regularity in phases at an automatic level. TD participants exhibited a P3B and an LPC in response to local and global facial emotion regularity, suggesting that the processing of abstract regularity is highly dependent on attention and conscious awareness of the stimulus. The absence of the P3B and LPC in SD suggests that the autistic brain cannot detect violations of the global facial regularity in a conscious manner. The second experiment used affective cores instead of facial images. The participants and paradigm were identical to those in the first experiment. The sequences were composed using consonant and dissonant chords. The experimental procedure and task were identical to those of the first experiment. The order of the two experiments was counterbalanced across participants. For both groups, variation of local emotion regularity elicited a mismatch negativity, followed by an N2. No let component was observed for both groups. However, only TD participants exhibited a let negativity in response to global music emotion regularity. This effect was distributed over the whole brain. Both groups showed comparable neural responses to variation of the local musical emotion regularity. The N2 might reflect attention-related mismatch detection of local emotional deviance. The let negativity is an ERP component often observed in memory tasks, so the absence of the let negativity in SD suggested that the autistic brain was unable to evaluate violations of the global emotion regularity in music based on the memory traces. In sum, we investigated how the autistic brain responds to local and global emotion regularities in facial and musical sequences. Results showed that in both facial and musical sequences, as the participants exhibited impaired brain responses to violations of the global but no loss of local emotion regularity. These findings suggest that the emotion regularity processing in individuals with ASD is modulated by the time scale of stimuli. I'd like to thank my collaborators from Shanghai Normal University and University of Reading. This study was supported by European Research Council Starting Grant and the National Natural Science Foundation of China. Thank you for your attention.